So hi everyone, welcome to Raaonline for another session. So in this short session, we will be seeing about bile salts. Okay, remember previous session I have discussed about the synthesis of cholesterol. During the synthesis of cholesterol, what I mentioned how the cholesterol is being dumped from your body. Remember, the only form of cholesterol excretion from your body is through the biliary canaliculi in the liver. That is the only form from where the cholesterol can be taken out of your body. So, bile acids and bile salts. For what these bile acids and bile salts are important? What is the significance of having this bile acids and bile salts? Okay. First, what is a bile acid and what is a bile salt? Acid has got a COOH group in it. So, in this case, this is one of the major component of bile. Hence, they are called as bile acids. What is the important raw material which is being required for synthesis of these bile acids? It is nothing but our cholesterol. Clear? And if this acids gets conjugated with various kinds of other components, okay, so something like amino acids, these are called as, they get converted into salts. So you call them as bile salts. So what exactly is the property of this bile acid and a bile salt? These are superpolar molecules. What do you mean by superpolar molecules? They have a very high negative charge. So what is the purpose of having such a very high negative charge in case of bile? The important property here is that the main role of them is to emulsify the fat and to form them into various kinds of small thing called as micelles, so that it could be presented in a much better way to the pancreatic lipase, which is there secreted from the pancreas. In this way, the digestion of lipids is being carried out in a efficient manner. Otherwise, remember, I discussed in the digestion and absorption of lipids, the chyle will be a big fatty chunk. Your chyle will be mostly the chyle will be more watery. This fatty chunk in water should be floating on the top, and the pancreatic lipase, which is being secreted from your pancreatic enzymes, will be there mixed in the chyle. So how can this pancreatic lipase go and digest the one which is on the top? That is quite not possible, right? Right. Now you need to break the big fatty chunks and you need to bring those fatty chunks inside the chyle, so that they can get acted upon by this pancreatic lipase, and they can get finally digested to form smaller particles which can be easily absorbed. So this thing is being carried out with the help of bile acids and bile salts. So what these thing does, these are, I told you before, these are superpolar molecules. So they easily go and encircle these fatty chunks. First, these fatty chunks will be encircled by phospholipids, and afterwards these bile acids comes and cover them. So because of their superpolar nature, what it does, it churns and it tries to break these fats. It has a detergent kind of an action. So in this way, the big fatty chunks can be broken down into smaller pieces, and because of this superpolar nature of this bile, which is now encircling these small fatty chunks, these small fatty chunks could be brought inside the chyle. It can get dissolved in the chyle. Okay, once it comes inside the chyle, with the help of co-lipase, they will be nicely presented to that of the pancreatic lipase, and the pancreatic lipase can act on this fats and it can break down these fats. So this is how a bile acids and bile salts has a significance in your body. Now in this session, what you do, we are going to see how this bile salt is being synthesized in your body. So remember, there are two important pathways for the synthesis of bile salts. So one is through a direct neutral pathway, and the another one, it is called as an acidic pathway. So the majority of the bile acids will be formed only through this neutral pathway, and remember, all these things are getting carried out, all these things are getting synthesized in your hepatocytes in the liver. Clear? So, what is required for the synthesis of bile acids? Cholesterol. Okay. So, cholesterol is nothing but it has got a sterol ring structure to which a OH group is attached at which carbon? At third carbon. Remember, and you have got a double bond at fifth and sixth position. So, this is what is a cholesterol molecule. Remember the structure of cholesterol, it has got 27 carbon atoms. Okay. So, this is the formula, structural formula for cholesterol. Clear? Now, this cholesterol, we will first see the neutral pathway. Now, this cholesterol will be acted upon by a cytochrome P450 enzyme, which is nothing but 7-alpha hydroxylase. It is a cytochrome P450 enzyme. Okay, seen in the endoplasmic reticulum. So, this what do you mean by hydroxylation? It is a monooxygenase. You are going to add what? You are going to add a OH group to this cholesterol molecule. Now, so from where you get this extra oxygen? I mean, like from where you get this energy? This can be got with the help of various kinds of reducing equivalents. In this case, the reducing equivalents is NADPH2. From where you can get this NADPH2? This NADPH2 can be synthesized from what? From HMP shunt. Also from the malic enzyme. You remember the fatty acid synthesis also requires this reducing equivalents in the form of NADPH2. You remember? Right. Now, this NADPH2 will provide these reducing equivalents so that this hydroxylation is being carried out, and what you do, you attach a OH group at the 7th position. You remember 5, 6 and 7. Okay.